For thousands of years, the human body has been an endless source of questions. Generations of scientists have devoted their lives to finding answers to these questions, leading to spectacular breakthroughs in our knowledge and in human health as a consequence. Still, from each answer, new questions arise, and the human body is now the ultimate and most ambitious frontier for scientific exploration. Like in any other kind of exploration, revolutionary tools and innovative strategies are needed to reach new horizons, such as those provided by Systemsx.ch, an unprecedented national research initiative created and funded by the Swiss government to revolutionize biology research, and as a result, boost knowledge of the human body and the basic mechanisms of life. This is the story of an intrepid Swiss initiative, of great intellectual adventures, and a surprising journey through the human body. Our eyes, our windows on the world, the most important connection between us and the world around us. They are the ideal gateway through which to enter the human body, the perfect start for our adventure. The human olfactory sense cannot compete with that of a dog. Our ears are not as sensitive as those of a cat, and we do not possess a sense based on ultrasounds as bats do. But our sight was designed by nature to be perfect for our needs. That's why scientists are working on strategies to decrease the possibility of losing it. But first, we need to understand more about how it works. To see an object, light needs to reflect off it and into our eyes. The light is focused on the retina, where different kinds of cells act as sensors, recording movements, colors, and shapes. The information then travels to the brain, which elaborates it. This is the moment at which we perceive we are seeing something: a car, food, a person we love. Michele Fischella completed his PhD in Basel, studying vision as part of one of the research projects financed by the SystemsX.ch initiative.
We can see thanks to at least 60 cell types in our eye. Each one of these cells is specialized in a specific role. And there are at least 7 different types of cells that are specialized in detecting movements. But the truth is that we know very little about what nerve cells do in our eye.
What is worse, we know almost nothing about how these various types of nerve cells interact with each other and the brain to result in vision. Knowing what each cell in our retina actually does in detail is the prerequisite not only for understanding how human eyes work, but also for finding cures for the diseases that put vision at risk. Above all, for diseases related to aging that are becoming more and more frequent as a result of increasing life expectancy. One example is maculopathy, which leads to the loss of central vision. This disease affected the artist Edgar Degas, and he dedicated himself to sculpture in the latter part of his life, since he could execute it using mainly his sense of touch. Maculopathy is expected to affect 196 million people worldwide by 2020. And according to estimates, it affects 1 in 5,000 in Switzerland. Another example is retinitis pigmentosa, which, on the contrary, impairs peripheral vision. It has been estimated that this affects 1 in 7,000 in Switzerland. These debilitating diseases currently have no cure.
We know that in all these illnesses, many cells are preserved. But to restore vision, we need to understand how these cells work in healthy and in disease conditions. And importantly, we need to know how to reactivate them.
In the retina, there are tens of thousands of cells, concentrated on an area of about 1 square millimeter. Researchers want to find out the role of each of them, and to this end, they need to record the activity of retinal nerve cells in a controlled environment. That's why they created this microchip, the most advanced of its kind. It allows the simultaneous detection of 2,700 nerve signals. For this, the researchers need a retina whose structure is comparable to that of a human, a mouse retina.
The mouse retina stays functional for several hours after that it has been removed from the eye, and it also reacts to light stimulation and movements. Now, this retina can be placed on the microchip that was developed thanks to the SystemsXch funding, and we can record the reaction to movement of each cell in the retina.
This kind of research needs technological know-how to develop cutting-edge tools like this microchip, and the collaboration between biologists, mathematicians, and informatics experts to deal with the huge amount of data that is collected. All of these efforts are working towards a very ambitious goal: becoming able to correct sight defects in the nerve cells themselves, in the retina, and maybe in future, even deep inside the brain, where images are processed. The brain, where the innermost essence of being human lies. The source of our thoughts, creativity, emotions, and the physical place where memories are created and stored. This is the next step in our journey. Memories are so important that it has been said that we are our memories. But what do we know about how we remember and forget? We know a lot, and at the same time, too little. For instance, we know that to deal with all our memories, we need to be able to forget. Indeed, only very remarkable events stay in our memories for the rest of our lives. Unusual events, such as a colorful event at the Zurich lakeside, can be remembered for a few days. But for the most part, our daily experiences are almost immediately forgotten. It appears paradoxical that forgetting is at least as important as learning itself. But hyperthymesia is an impairment of the ability to forget, which illustrates just how important forgetting really is. The first person in the world to have been diagnosed with hyperthymesia was an American woman. Like other patients with her condition, she can remember what happened on each given day for decades. Even tiny details, such as weather conditions. But this is too much. A relentless flow of memories seems to overwhelm her. She has been reported to have several phobias and is constantly trying to keep order amongst her objects as well as in her thoughts. So, how do we forget? Just like everything that is very important for survival, memory seems to have evolved very early in living organisms, and not to have changed much over the course of evolution. That's why, in order to understand how we forget, it is very useful to find out how they forget. Fruit flies. They're at the core of the SystemsX.ch research project led by Simon Sprecher in Fribourg.
There are billions of nerve cells in the brain of humans, while there are only a few thousands in the brain of insects. Still, we share a lot of things with insects and all other animals, the genes that allow us to remember and forget. Therefore, we believe that the cellular mechanisms are the same in humans and in insects, and we want to identify those which are responsible for forgetting.
Researchers have spotted over 100 genes that are involved in learning. Now, to understand which other genes are only involved in forgetting, they alter the activity of each one of them, one by one, using special techniques. Then they look at how the alterations influence how the flies forget. These are very special flies. They have learned through training that a given smell leads to food, while another is associated with a tiny but unpleasant electric shock. After the training, researchers introduce the flies into a tube and leave them free to choose a direction. Those who have not forgotten the training fly towards the smell associated with food. The others fly randomly, with some of them eventually ending up in the direction of the smell associated with unpleasant sensations.
Using a super-resolution microscope allows us to even see what happens in nerve cells, which connections are stronger or weaker in flies who more easily forget. In this way, we can not only identify the genes which are involved in forgetting, but also to see their effect.
Understanding how forgetting works is also key to finding ways to identify the emergence of neurodegenerative disorders that can cause widespread brain damage and affect behavior. Forgetting is natural, but when something in the brain goes wrong, it becomes a red alert signaling something grave, a neurodegenerative disease. Loss of memory is indeed a distinctive feature of diseases such as Alzheimer's, but also of CJD, or Creutzfeldt-Jakob disease, the infamous human form of so-called mad cow disease. CJD is caused by prions, a type of protein that was identified as an infectious agent in 1982. Since then, it has been discovered that prions not only play a role in the development of diseases such as Alzheimer and Parkinson, but they are also responsible for other illnesses so horrific that they are reminiscent of an ancient curse, like the so-called fatal familial insomnia. The first known patient of this terrible condition was Giacomo, a man born near Venice in 1791. When he was 45, Giacomo suddenly stopped being able to sleep. Days and nights passed in an endless stream of waking nightmares. He was trembling, delirious, stricken with dementia. Finally, after months of tortures, he died. But the gene responsible for all this had already been inherited by some of his children. One generation after another, it was passed down. Giacomo's family is one of the few tens of families in the world in which the prion alterations that lead to fatal familial insomnia have been detected. But what else is due to prions? And why has evolution allowed them to exist in our body? In spite of decades of studies, prions are still relatively mysterious molecules. Adriano Aguzzi, working in Zurich, has devoted his working life to the study of the nature and function of prions, achieving spectacular results. prions are among the thousands of different proteins normally produced by our bodies in healthy conditions. The problems begin if they change their three-dimensional conformation and assume a pathological shape. In this form, they induce other prions to adopt this wrong shape. These pathological prions then pile up on the surface of the brain, killing the cells that transmit nervous signals and leading to a devastating disease.
If our body makes this prion protein, there must be a good reason, because the prion protein is potentially extremely dangerous. Therefore, if it wouldn't have an important physiological function, it would disappear in the course of evolution. So for the past 20 years, we have investigated in depth whether what might be this reason. And what we have found is that the prion protein is expressed by nerve cells, and it helps maintaining the health of the sheaths that envelope the nerve cells. And in its absence, these sheaths become sick, and this provokes a disease.
In other words, prions seem at first glance to be an innocuous bunch of Dr. Jekylls. Usually, they quietly go about their useful jobs inside our bodies throughout our lives. But sometimes, they suddenly turn into Mr. Hyde. And if that happens, they can eventually kill us.
To cure and possibly even to prevent prion diseases, we need to exactly understand how prions are built within the cells, what are the stimuli that lead to their production, and how this production is regulated.
To find answers to these very complex questions, Aguzzi's group conceived a project of titanic proportions, carried out with the support of Systemsx.ch.
In every cell of our body, there are more than 25,000 genes. And modern technologies allow us to switch off every single one of these genes. And what we are trying to do is to understand, upon switching off which gene the cell loses the capability to produce prions.
Such an enormous task would not even have been conceivable with the instruments available only a few years ago.
It's a complete change of perspective. We do not yet know what we are going to find. But if we find something, and I'm pretty convinced that we will, this will be completely unexpected, and it will open entirely new horizons for our research.
Since Galileo, the scientific method has consisted in formulating a hypothesis that must be verified with experiments. It is a bit like guessing where treasure might be buried in the desert, and then going out and digging for it to find out. By contrast, the large-scale screening conceived by Aguzzi's group is like combing through the whole Sahara and examining what's left in the sieve. So far, Aguzzi's group has screened about 6,000 genes. And just as it is often the case that fortune favors the bold, they have found something interesting.
Thanks to our project, we have already found one gene which controls the production of the normal prion protein. And what is most exciting is that this gene can be controlled itself using relatively regular drugs. So the hope would be that on the basis of these findings, that we might actually even propose a therapy for Creutzfeldt-Jakob disease and similar diseases. And it even goes further than that, because at the molecular level, Creutzfeldt-Jakob disease is very similar to other diseases which are extremely common, such as, for example, Alzheimer's disease and Parkinson's disease. So on the long run, my greatest hope would be that the discoveries that we are making in the frame of prion diseases may even be applicable to much more common diseases.
Nerve cells, muscle cells, skin cells, fat cells. The body is made up of about 200 different kinds of cells. Some of these are very special. They are called adult stem cells and can generate mature cells of different types in a specific tissue or organ. In the bone marrow, there are adult stem cells called hematopoietic stem cells. They maintain the blood-forming system over the entire lifetime of the organism, generating all the different types of blood cells, including white blood cells involved in immunity and erythrocytes responsible for delivering oxygen to different tissues. Hematopoietic stem cells provided by a donor form the basis of life-saving treatments for those affected by tumors, such as leukemia, by metabolic or immune system diseases, and many others. But to treat these conditions, there is also another option, using stem cells extracted from the umbilical cord of a newborn. Weijia Wang in Basel, whose research is supported by SystemsX.ch, is working on understanding how cord blood stem cells could be made to multiply in laboratory conditions without losing their identities as they would naturally do.
These cells receive signals from the outside, process the information, and react in response. They are like factories, receiving orders and responding accordingly. They make decisions all the time. We need to identify the signals that can make them change and lose their characteristics, as well as those that can make them keep their qualities while multiplying.
Although Weijia Wang's research is revealing the secrets of cord blood stem cells, her discoveries will contribute to a better understanding of all kinds of stem cells, which are a never-ending source of questions in modern biology. Scientists want to learn about them because their potential for application in the treatment of disease is immense. The possibility of influencing the behavior of stem cells would open completely new horizons for biology. In the little zebrafish, stem cells help the brain regenerate after an injury. Will we ever be able to induce something similar in humans to regenerate our damaged organs? And more, to what extent will we be able to safely manipulate our cells? At the University of Zurich, Lukas Pelkmans is a strong advocate of quantitative cell biology.
Today we can only imagine that in the future we are able to control and change the fate of our cells, our organs, and even our entire organism. But as a first step towards that direction, we need to understand how biological systems work, how they generate complexity. This is exactly the aim of the Systemsxch initiative. To move away biological research focusing on a single molecule such as DNA or protein towards research that tries to study multiple molecules together, such as DNA, protein, and also many other components. How they together generate complexity, and how from that complexity properties emerge that could not have been envisaged without looking at the whole picture.
To understand what Lukas Pelkmans means, let's think for a minute of the human body as a termite nest. Just like the human body, this mound is one of nature's highly complex inventions. Its sophisticated structure protects it from hostile weather and enemies. Inside, different spaces are dedicated to raising larvae, storing food, hosting the queen, and the corridors are exactly the right size in order to be both accessible and easy to defend. Now, if we were to focus on an isolated termite, no matter how carefully you look at this single insect, you will never be able to figure out this.
Pretty much as we can understand many details of the physiology of a single termite, we can understand a lot about a single gene and its product. But this one gene can have a pronounced effect in one person without having any effect in another person. It all depends on its specific context. And for this reason, we need to understand how genes interact within the complex system of a single cell. We need to understand the whole system to understand the functioning of a single gene.
What is very interesting is that doing systems biology also means taking chance into consideration. Chance can play a role in why and how a cell lives its life, or dies, or becomes ill.
The good news is that in spite of this chance, of these probabilities, we can still predict the outcome of molecular processes, or control the fate of individual cells, predict illness or the reaction to a drug. In order to achieve that, however, we need to integrate huge amounts of quantitative data. Fortunately, today we have unprecedented computational tools and technologies, and we can carry out experiments, thousands of experiments simultaneously.
Understanding the emergence of an illness or the effect of a drug sometimes extends beyond fighting against diseases that develop within our own bodies, such as cancer or heart disease, and requires facing old enemies, well-known killers with a highly sophisticated capacity to interact with our bodies and take control, pathogenic microbes. Among them, the pathogenic bacteria that we are about to encounter in the lungs. Over the last century, the threat to human health posed by bacteria seemed to be coming to an end thanks to the use of antibiotics. Drugs with the seemingly miraculous power to stop bacteria. But what had been totally underestimated is that bacteria have a unique capacity to change, to evolve new characteristics, helping them survive in hostile environments. Because of this ability, bacteria can become resistant to the drugs, and they fight back. Among antibiotic-resistant bacteria, there is particular concern about those responsible for tuberculosis, which mainly infect the lungs. Perceiving the problem of tuberculosis as far removed from our everyday lives would be a mistake. In fact, it recently turned up right on our doorstep in Basel. Here, in 2010, an apparently healthy 35-year-old man was walking around. He had just arrived in Switzerland from Tibet. But somewhere on his long journey, he had picked something up that was now multiplying in his body, a form of Mycobacterium tuberculosis. When he was diagnosed with the illness, doctors realized that the bacteria that infected him were resistant to all of the currently used antibiotics. Thankfully, a new treatment was being developed, and given the gravity of the case, the doctors decided to try it out as the last hope to treat the patient. In March 2013, after more than 2 years of treatment and a long hospitalization in this security room in a high-altitude clinic in the Alps, he seemed to have defeated the disease. But a few months later, he fell ill again, and this time, the microbes had even developed resistance to the new drug. Once again, he received another experimental treatment. Almost incredibly, the bacteria also developed resistance to this drug. To decrease the level of infection, it was necessary to remove part of his lungs. Finally, in 2015, after almost 5 years of invasive treatments, he was declared cured. Sébastien Gagneux in Basel was unable to overlook this story. He leads a project financed by Systemsx.ch, which is focused on understanding the development of antibiotic resistance in Mycobacterium tuberculosis.
The scary story of this patient tells us something. Although today, the large majority of TB patients can usually be treated with standard treatment, these super-resistant bacteria can emerge even during the treatment of an individual patient. This puts lives at risk.
In Switzerland, around 500 new tuberculosis cases are recorded every year. And worldwide, this number is more than 10 million, leading to 1.8 million deaths. It has been estimated that a further 2 billion people, that's almost a quarter of the planet's inhabitants, host the bacteria in their bodies.
Although the problem of drug resistance has been known for a long time, we still not fully understand how it actually works.
To work with dangerous microbes, special precautions have to be taken. This is a biosecurity laboratory. Researchers are required to wear odd-looking overalls for protection. A pressure gradient between the lab and the outside assures that no air can get out of the laboratory. And every piece of clothing or object that has been inside must be treated at a very high temperature before eventually being taken out to be discarded.
Here we study how bacteria evolve both in presence and absence of antibiotics. We also study how new resistance forms and evolves, and what kind of substances the bacteria produce when they become resistant. Our final goal is to find a way of identifying these bacteria with the highest probability of becoming super resistant and highly transmissible. Ideally, this would allow us to prevent severe cases of tuberculosis like the patient from Tibet in the future.
Knowledge is power in the war against pathogenic microbes, as in any other enterprise. What is at stake is our ability to treat diseases that have the potential to kill millions. Leaving the lungs and the bacteria that can infect them, let's move down into the body, towards the belly, where unwelcome fat tends easily to accumulate. Love handles have a cute name and a bad reputation. They have been designed by evolution to serve as an energy supply to be used when food is scarce or when extra energy is required. For thousands of years, our starving ancestors considered them as an alluring, almost unattainable mark of freedom from hunger. But today's society is vehemently against fat, and we know that weighing too much puts our health at risk. But let's look at fat from another angle. Without it, we simply would not exist. From a scientific point of view, what we call fat corresponds to a variety of molecules named lipids. In Lausanne, Guizou van der Goot is leading a project funded by SystemsX.ch, which aims to gain a better understanding of the large part of us that is made up by lipids. Although lipids tend to be identified with that surplus weight that we'd rather not have, they are actually everywhere in our bodies.
Indeed, each of our cells is like a little bag, and the wall of this little bag is formed of lipids. But in order to communicate between the inside and the outside of the cells, In this lipid layer, there are proteins that allow messages or signals to get in. Then inside the cell, there are also chambers, and these chambers are again surrounded by lipids. The biggest of these chambers is the nucleus. This is where our DNA is. And there are also all kinds of other little chambers, and one of them, for example, contains the fat. All our cells have fat, some very little, and others that we call fat cells have a huge amount of lipids, and they contain in particular cholesterol and triglycerides.
Cholesterol, one of the most fundamental molecules in living organisms. Adults have about 150 grams of cholesterol on average. That's more or less the weight of this soap, and almost all of it is necessary. Cholesterol forms part of the covering of nerve cells that allows them to transmit signals. Our body needs it to produce vitamin D and avoid diseases such as rickets. And it is the basic ingredient for many hormones, including sex hormones.
So indeed, cholesterol is a very important molecule, and therefore cells have two ways to get it. One is to get it from the diet, and the other one is to make it themselves. So it's a very complex molecule, so there are 20 steps to make it. We know what these 20 steps are. What we don't know is how cells determine whether they take it from the diet or whether they make it themselves and how fast they make it. And also we don't know how cells completely get rid of it when they have too much.
Lipids exhibit complex molecular behavior and interactions with other components of the organism.
So lipids in a membrane behave as a community. They influence each other, they move in a synchronized way, and then they influence the proteins that are inside of this membrane. And vice versa, the proteins will influence the lipids, and also the inside and the outside will influence the lipids.
To understand which genes control the variability of different kinds of lipids and how this variability changes according to the needs of the cell, scientists are switching off each one of the roughly 25,000 genes, one by one, in order to study the effect on each of the roughly 1,000 lipids that are in our cells. This means taking 25 million measurements.
To analyze all these results, we have to use mathematics and high computational power. And this is typically the kind of approach that has been prone by SystemsX.
The kind of studies carried out by Gizou van der Goot and her collaborators are of fundamental importance to understanding not only how our bodies work and stay healthy, but also to understanding what happens when something goes wrong. If this happens, it can lead to metabolic diseases, such as diabetes and obesity, that at present are among the most common diseases in the world. Would you ever have guessed that lipids are so important for our existence? Scientists are constantly broadening our awareness of the importance of our body's constituent parts. And even the most neglected can turn out to play a pivotal role in the organism as part of a complex system. But maybe none of them is so unexpected as the one we are about to approach as we make our way down into the gut. What we are about to learn might give us a new perspective on existential questions such as, who are we? Surprisingly enough, we should actually think of ourselves as colonies composed of billions of different living things. Indeed, in our bodies, there are as many bacteria as human cells. And the number of bacterial genes inside us is about 100 times the number of human genes. The next time you weigh yourself, consider the fact that at least 1 kilo is due to the microbes living in your gut, called gut microbiota. These tiny inhabitants play a vital role, helping us digest food to obtain energy, producing vitamins, breaking down toxins, and so on. Andrew McPherson in Bern has devoted his life to the study of this overlooked part of ourselves, and he is currently leading a project funded by Systemsx.ch.
Our bodies are populated by enormous numbers of bacteria. These live in the intestine and on many body surfaces, and often do us absolutely no harm. Depending on which sort of bacteria are present in our bodies, we can be healthy or they can cause disease. So examples of these diseases are diabetes or depression, but many other conditions are also influenced. If we are healthy, then part of our health is due to the exchange of molecules between these different bacteria and the rest of our bodies.
Basically, there are no two people in the world with identical gut microbiota. It develops during childhood according to our environment. And although it will resemble those of our parents or the people who live with us when we are young, it has its own unique personal character.
Not all the bacteria that are present in nature can actually colonize the intestine, although the number present in the intestine is rather large, about potentially 1,000 different species. Of these, each of us has about 100 different species present in our insides.
If the human body were planet Earth, the gut would be the rainforests. Here, thousands of different organisms live harmoniously. The system might look chaotic from the outside, but it is not. At a closer look, each species has its own role, and there are almost no unexploited resources. That is why immigrant species have a difficult job of integrating. But how does our inner rainforest work?
Effectively, we've only just started the exploration of the gut microbiota. As part of the Systemsxch project, we are trying to understand how the exchange of molecules between the different sorts, the different species that exist in the gut microbiota works, and how the exchange of molecules between the different species of gut microbiota and the rest of the body influences our disease or health status.
The gut microbiota is located in the mucus that lines the internal walls of the gut. To study it, researchers collect samples by endoscopy. Then a team of biologists and mathematicians works to develop models that can predict the interaction of the bacteria in the different samples. And their results are compared with what is observed in reality. One important goal of these studies is to help patients with inflammatory intestinal diseases. The number of bacteria species in their gut is too low. The rainforest turns to desert. And just as in natural environments, this drastic change is very difficult to reverse.
Unfortunately, in many clinical situations, it's rather difficult to be able to introduce good bacteria to restore health in a therapy context. The reason for this is probably that it is such a complex situation in the intestine that it's difficult for us to decide exactly which bacteria we should use as therapy for patients. Once we know exactly who our gut inhabitants are and how they relate to each other and to our bodies, we will be able to start to design therapies and to use the clinical situation in order to improve patients' health.
Microbes actively inhabit not only the gut, but also the lungs, the throat, and they thrive on the 2 square meters of skin covering our bodies. Between 1 and 10 million such creatures live on each square centimeter of skin. And the more we know about them, the more we can uncover the specific role they play in our health. Healthy skin is not just a matter of appearance, but something centrally important for human health. It is our first-line defense against the outside world. It is what we see when we look at other people and what we feel when we touch them. But sometimes it can harbor life-threatening disease, a melanoma. Melanomas are malignant tumors originating from the degeneration of melanocytes, the cells that produce the pigment that gives our skin color. In Switzerland, there are over 12,000 new cases of melanoma every year. Risk of developing the disease is high among those with a pale skin tone, like the majority of the Swiss population, as it offers little protection against dangerous UV solar radiation. The national passion for outdoor activities doesn't improve the situation. Olivier Michelin is leading a Systemsx.ch research project in Lausanne, aimed at revealing the different ways metastatic melanomas adapt to current therapies and limit their long-term efficacy.
Metastatic melanoma is a very aggressive disease. With current treatment options, 20% of the patient can be stabilized and doing well without symptoms for at least 10 years. With newer regimens, we are moving to 40%, and with current research, we hope to move the bar to 80, hopefully 100% in the coming years.
Up to the end of the last century, tumors were mainly classified and treated according to the principal organ involved: breast, prostate, lungs, and so on. But over the last 15 years, it has become more and more clear that each tumor is a highly complex agglomeration of different cells, not all of them tumorous. To some extent, every tumor is unique. This complexity explains why the same therapy does not always give the same result in two different patients.
In order to improve the number of patients who can benefit from treatment, we need to be able to understand the disease in molecular detail. We need to be able to understand why the disease in a given patient is responding, whereas the same disease in another patient does not respond to treatment. We also need to be able to understand why a disease is responding initially in a patient and then is progressing. So we need to be able to analyze the molecular pattern, the mechanism by which the disease is now able to resist therapy, to bring further discovery and new therapeutic solution for the patients.
Olivier Michelin and his collaborators study samples, each containing about 1,000 cells.
We analyze gene use by single cell obtained from the tumor. We can analyze the tumor before treatment or after treatment. We see very big differences in the gene used before and after, which help us design subsequent therapy.
In other words, the cellular composition of a tumor changes under therapy. Some dangerous cells are usually killed, but in many patients, some of them survive or even go on to develop resistance.
With this project, we are learning a lot about melanoma. This is research at the moment, but each time we understand a new mechanism, we are getting closer to personalizing the treatment for that patient.
In the fight against cancer, scientists are increasingly able to confront each individual tumor with a combination of different strategies that include these standard ones, such as chemotherapy, but also the more advanced ones, such as immunotherapy, which are increasingly targeted to hit each cell's Achilles heel. This is a goal that could never have been reached without a systems approach to biology. Indeed, in order to understand how a tumor works, researchers must study it as a whole, focusing on the interactions between its components. This is not only the case for tumors, but basically for every single part of the body. Rudi Ebersold is a renowned Swiss biologist and one of the main supporters of the need to move towards system-focused biological research.
The knowledge gained over the last 20 to 30 years, specifically knowledge gained in the analysis of genes and genomes, have been enormously important for biology, and the significance can really not be overestimated. We have also learned that the information of genomes by themselves are not sufficient to explain the functioning of biological processes. For instance, it has been observed that different mutations, different changes in the genome expressed in different individuals have different effects. We therefore had to abandon the idea that knowledge of the whole genome and the products it generates would be sufficient for the understanding of biological processes and eventually the functioning of the human body.
In the 80s and 90s, one of the most advanced frontiers in biology was the study and sequencing of the human genome. The achievement of its first draft was enthusiastically announced by both Bill Clinton and Tony Blair on the 26th of June, 2000. At that time, large international collaborations were established. Some countries decided not to take part in the venture, assuming that they would be able to join in later and use the information for their research. Switzerland was among those that took the decision not to participate in the human genome endeavor.
In my opinion, this was a big mistake. In life science research, it is not just the results that are being communicated, but also the process that leads to the generation of these results. Consequently, the countries that were in the genome project early on, they not only had first insights into the results, but they were also learning the processes that are the basis of genomics research. This persisted in the academic research and biotech industry culture, in that these countries are still leading in that field today. In contrast, Switzerland, while generally very strong in life science research, never attained a worldwide leading position because we missed the initial genomics trend. With the emergence of systems biology, the decision was taken to create programs that would allow Swiss science to be at the forefront worldwide in the field of systems biology. This was the reason that Systemsxch was created in 2008 for the benefit of the population and of the country as a whole.
But now it's time to go back to our skin, because there's something we might have overlooked. Just how incredible it is. Our skin not only isolates our inner organs from the surrounding environment, but it also allows the exchange of heat, helping the body maintain its ideal temperature. Human skin is so much more than a simple wrapper. It is a highly complex organ composed of many different cells. And in fact, it is the largest organ we possess. How does this skin of this reptile or of this mammal compare to our own? Do they look very different from ours? On the surface they do, but surprisingly enough, there is a lot that we have in common. Michel Milinkovic in Geneva is leading a project financed by SystemsX.ch, which aims to understand how the amazing variety of skin types in nature arises.
There is no doubt that we can understand the development and function of the skin, including the human skin, only if we compare the diversity and complexity of skin across species. For example, in my research group, we have demonstrated that the production of hair in mammals, and of course in humans as well, but also the development of feathers in birds and of scales in reptiles, require the formation of local thickenings of the embryonic skin made of specific cells that express in all species the same set of genes. And if one of these genes doesn't work, then the embryo develops into a hairless mammal or a scaleless reptile, like in these scaleless lizards.
In humans, if just one of the genes studied by Michel Milinkovic does not work properly, the results can be surprising. This is the case for the actor Michael Berryman, who has very reduced hair, fingernails, teeth, and sweat glands. More or less the same happens in bearded dragons, a species of Australian lizard. Usually they have scales all over their bodies, including spines on the head and on their flanks. But if one gene is mutated, the scales and spines grow much smaller, and all scales and spines disappear in the offspring of two mutated parents. That is what happened with this individual, whose skin is incredibly smooth. Although genes play a fundamental role, how skin looks might also be affected by a number of other factors, including the shape of the cells and the compression or tension to which they are subjected.
We model the biological and physical processes that generate the skin shape and color in different species. This is why we are using a systems biology approach. Not only do we rely on skills and tools typical of biology and genetics, but also on those from physics, computer science, and mathematics.
The studies carried out in this lab are an essential step towards being able to reproduce fully functional skin, complete with hair and maybe even other parts, such as glands and touch sensors. Something that today is not yet achievable, but which would be a huge step forward in helping those who have suffered severe burns. Our skin has another, not always desirable, characteristic. It easily shows our age. Unavoidably, our bodies age. Still, some of us seem to age more slowly and stay healthy for longer. Like Jean-Louise Calment, who is so far the longest living person ever recorded. At the age of 85, she started fencing. And she was still cycling until she reached 100 years old. She passed away at the age of 122 years and 164 days. Bart de Planck, who is based in Lausanne and is leading a SystemsX.ch project focused on aging, is very interested in discovering the secrets behind her long and healthy life.
Living a long and healthy life is a matter of luck, of lifestyle, and of genes. Now, we all know we can't control fate, but knowing the function of genes that are linked to aging And knowing which versions we have of these genes in our genome can really have a large impact, even to the point where it may allow us eventually to become healthy centenarians.
Bart de Blanke's research is aimed at identifying the effects of genes related to aging in the complex environments of cells, organs, and organisms as a whole.
We all can appreciate that making certain lifestyle changes can really have an important impact on our health. For example, if you know that you have high cholesterol, it's probably best to reduce your intake of greasy foods. But that said, there seems to be some people in the world that benefit from a certain genetic makeup that protects them from a very harmful lifestyle. For example, there's some people that smoke all of their lives and yet they never develop lung cancer. And by the way, this was true for Louise Calment as well. And so this is really a mystery. We really want to understand how these genes in this genetic makeup act. We want to understand which versions of your genes you have, either good or bad, and so that we can either promote or limit their impact. And then we want to change your lifestyle or even invoke a certain treatment such that it can have a lasting impact on your health.
So far, scientists have identified 30 genes that are expected to be key players in human longevity. The next step now is to understand their behavior and effects inside cells. In humans, this is an almost impossible task because we have a long lifespan and our environment exerts its effects on us for decades in a very diverse way according to what we eat, what we breathe, what we do, and where we go.
But luckily we can work with animal models, since animals such as mice, but even flies or worms, have genes, surprisingly enough, similar to humans that also affect aging. And these animals we can raise in very standardized and controlled environments. And that makes it much easier to study the role of a gene that is linked to aging.
For instance, these two worms are the same age, but the one on the left looks much more active. This is because it got the lucky version of a gene that influences its overall fitness.
As part of our project, our collaborators are creating very complex mathematical models. And this helps us to understand how a gene interacts with other genes or even with environmental factors. And then we have to study data collected on real people to see how the predictions and hypotheses that come forth from these complex mathematical models can be confirmed in the real world.
Researchers think that in a few years, it will be possible for each of us to have a complete fingerprint, describing which form of the genes related to aging we possess. This is likely to totally change our perception of getting old, transforming it into something much more under our control. The human body has been a black box for millennia. Over all this time, we've only been aware of what it displayed: illness or health, life or death. But then, very slowly, we started to learn more, and we gained a remarkable understanding of ourselves. But we know we need to go further. We must understand what is going on inside each one of us, because each of us is unique and has his or her own needs. Ralph Eichler is a physicist and has been one of the main proponents of the SystemsX.ch initiative since 2007, when he was the ETH president and the first chair of the board of directors of SystemsX.ch.
To reach these new ambitious goals, biology needs a breakthrough. For this reason, it is moving away from qualitative observations to quantitative measurements, precise models, computer simulations, which led to new understanding and descriptions of biological systems as a whole. This kind of comprehension is reached through the collaboration of scientists of different fields. In the area of system biology, the observation of living organisms benefits more than ever from the knowledge of physicists, chemists, engineers, computer scientists and mathematicians. On the other hand, the study of biological functions certainly will lead to new engineering solutions which will find application in your daily life. As an example, to understand how our brain recognizes images may lead to new electronic circuits which will be implemented in your new mobile phone. SystemsX.ch as a project is a true win-win enterprise.
Overall, the Systemsx.ch initiative has financed over 250 projects involving more than 2,100 scientists. Although this documentary is focused on studies related to the human body, the majority of the projects supported by Systemsx.ch involve basic research and are only remotely related to our species. What is also important is that Systemsx.ch has contributed to changing the vision of a new generation of researchers through financial support specifically designed for young scientists. This is the future of life science, and through the Systemsx.ch initiative, Switzerland paves the way in exactly the right direction for the benefit of its citizens and all human beings.
I think that this project will have long-lasting effects for Switzerland. It will beneficially affect the research in the public as well as in the private sector.
My interdisciplinary PhD project enabled me to approach research questions from a different angle and to think differently about my career and my future.
Our Systemsx.ch project has allowed us to extract useful information from a huge set of data thanks to the combination of experimental biology, mathematics and modeling.
The way we are conducting our research has changed profoundly over the last several years. We need new instruments, new methods, new strategies. And in this context, Systemsxch has been extremely useful. It has given us the right push at the right moment.
Through SystemsX, I got a unique learning opportunity in a multidisciplinary research environment. This will help me a lot in achieving my career goal, which is to establish my own research group using the tools and approaches that I've learned here.
Our SystemsX project is helping to answer fundamental questions about tuberculosis, eventually leading to better prevention of the spread of this dangerous pathogen globally and also here in Switzerland.
SystemsX has really changed the way we do this type of research, and I cannot imagine we could go back to the way we did it before.
The collaboration in our SystemsX project has been absolutely transformative. It's given us the opportunity to understand our inner world of intestinal bacteria, how these influence our bodies, and how we can change these interactions in order to improve our health.
Systemsx was a very ambitious and very brave initiative. At the beginning, many thought that this would not work, and indeed it took a long time. But now when we look at what has been possible through this initiative, we realize that we were on the right track.
By bringing together in the same research group cell biologists, geneticists, physicists, computer scientists and mathematicians, we can start to answer questions that we could not answer in the past, opening of course new avenues of research.
The substantial network of researchers working in different fields that has established itself thanks to Systemsx.ch will for sure continue to collaborate, to work together, even after the initiative will have ended.
Thanks to Systemsx.ch, Switzerland is now a world leader in systems biology.